what's this? Well, this one. After anesthesia. Oh, yeah.
Oh, that's fine.
Okay, you're still missing those stuff.
Oh.
That's what we're missing. Missing anything as well. Okay.
And you could remind presenters, people in the audience, if they have a question, to go to the microphones.
Yeah. Okay, we're gonna do questions after each speaker.
After each speaker?
Yeah.
Okay.
I don't know, I like that format better. If that's okay with you guys. We have 30 minutes, right? Here's what we do. All right. Yeah, do you guys want to just hang out here, stay up here, or do you want to come from the audience?
Doesn't matter.
Let's stay on the stage.
If they're all up here, it doesn't take time
Yeah,
for them
that's
to.
about less time.
Okay, so O is the only person. O Hamilton.
Yeah.
Hamilton O. Sorry.
So.
And they aren't here.
Yeah.
We'll start soon.
We can start this 2 days past 1.
Yeah.
We're gonna
Okay.
we can switch the order. Why not? Let's get in the
Yeah, we can do Hamilton last. Okay.
Just in order.
order. That's okay.
We're going to do order, but we might skip Hamilton and go to the end. Put him at the end. So, that doesn't, yeah.
Yeah, okay.
But otherwise, we'll preserve the order.
Okay.
And make sure I got people's names. How do you say it?
Lou? It's Lou.
Lou?
Yes.
So I just introduce you as Lou?
Yes.
Okay. Introduce Steve?
Yes. Hamilton is here, right?
I think we should get started.
All right, you want me to get started? Okay, hi everybody. Thank you for being here. The title of this session, just to make sure we're all in the right place, and that I'm in the right place, is Proteomic Signature Prediction and Mechanistic Insights into Cognitive Decline. So thank you for being here. My name is Keenan Walker. I'm a senior investigator in the NIA Intramural Research Program, and this is my co-chair, Teresa Tien, who's a staff scientist in the NIA Intramural Research Program as well. And we have the honor of introducing a series of great talks for you this morning. So we're going to go ahead and kick it off, and I'm going to let Teresa make the first introduction.
Great. Thank you for being here. So the first talk is given by Dr. Federica Anastasi. Her title is Predicting Cognitive Trajectories in Asymptomatic Individuals at Risk of AD.
And just to let everybody know, right before we get started, we're going to do questions after each speaker. We're going to do 10 minutes for talks, 5 minutes, hopefully, for questions. So come to the mic after each speaker if you have a question. Otherwise, I'll read any online questions. All right, go ahead.
Thanks so much for the introduction. I would like to thank also the Alzheimer's Association for the travel fellowship that allows me to present these results today. I have nothing to disclose. Alzheimer's disease is a highly heterogeneous disease, and we are seeing substantial inter-individual differences in both the disease onset and disease progression. And while we now have biomarkers that can detect with high accuracy the presence of amyloid and tau pathology, we cannot say among those with pathology who is going to decline when and how fast. And this suggests the presence of molecular mechanisms that goes beyond amyloid and tau. And understanding this mechanism is of key importance for both identifying prognostic biomarkers that can be used for clinical trial recruitment or risk stratification, but also to identify novel therapeutic targets. So in this project, we leverage novel advances in antibody and aptamer-based proteomic technology that now enables the measurement of thousands of proteins, giving a real-time snapshot on the molecular processes. And the preclinical phase of Alzheimer's disease, that is the most promising window for early intervention, is still remain poorly understood in terms of heterogeneity in cognitive trajectories in this phase, as well as sex differences in the mechanism associated with cognitive changes. So our hypothesis is that heterogeneity in cognitive changes is linked to distinct molecular mechanisms that are already detectable in preclinical Alzheimer's disease, and that these mechanisms may differ by sex. So we define two different objectives. The first is to identify plasma proteins linked to cognitive changes in cognitively unimpaired individuals and to compare this molecular signature between amyloid positive and amyloid negative individuals. And as a second objective, within the preclinical AD, so within the amyloid-positive individuals, we wanted to explore whether differences in cognitive trajectories are associated with sex-specific molecular signatures. In this study, we included 383 participants of the Alpha Plus cohort, that is part of the Alpha study of the Barcelona Beta Brain Research Center. And all the participants are cognitively unimpaired and had higher risk of Alzheimer's disease. All of them had plasma proteomics at baseline measured both by Olink and SomaScan technologies, as well as AD core biomarkers, and they've been followed up every 3 years for a total of more of 10 years of follow-up. And in particular, in this project, we are focusing on the preclinical Alzheimer cognitive composite that is particularly sensitive for the preclinical early subtle cognitive changes. So I would like to stress here the fact that amyloid plus, the alpha plus individuals are cognitively unimpaired. Being cognitively unimpaired, we already see very heterogeneous cognitive trajectories during time, with a significant difference between amyloid positive and amyloid negative individuals, with amyloid related already to the loss of learning effect a long time. And as you can see in the table, we are seeing a significant difference between the amyloid negative and positive group in terms of age, with the amyloid positive group slightly older, and in terms of ApoE4 carriership, with the amyloid positive groups having a higher percentage of ApoE4 carriers. So to identify the proteins at baseline that are associated with longitudinal cognitive change, we use a linear mixed effect model using 4 time points of PAC. We included a random intercept and a random slope for accounting for inter-individual variability, and we corrected the model by age, sex, and years of education. So to address the first objective, we first stratified the analysis by amyloid status, and to address the second objectives within the preclinical AD group, so within the amyloid-positive individual, we stratified the analysis by sex. We then performed a pathway enrichment analysis on the significant proteins, and then finally a cross-platform validation to identify those common significant proteins that have the same effect direction in the same group. I'm going to show you now the results of the first part of the study in which we stratified the analysis by amyloid status. As you can see in the volcano plots, you see the proteins that are associated with cognitive changes. And to help you interpret the plot, in the x-axis, you find the effect sizes of the interaction of protein with time. And a positive effect means that higher the protein level, better the cognitive changes during time, while negative effect means higher the protein level at baseline, worse the prognosis we have. And you can see those results for the amyloid-positive group for both Olink and SomaScan. We also repeated the analysis in the amyloid-negative group. I'm not going to show you now the volcano plots for a matter of time, but what is important here is that we found a very small overlap of the significant proteins between amyloid-positive and amyloid-negative groups. So this suggests already that the signatures associated with cognitive changes are different between groups and potentially also the mechanism. And to understand better this, we perform an enrichment analysis on the significant proteins, and we identified that the proteins that are associated positively with cognitive changes over time in the amyloid-negative group are enriched by inflammatory response and response to external stimulus. When we repeated this analysis on the amyloid-positive group, what we identified is that proteins that are associated negatively with cognitive changes, so the higher the protein, the worse the prognosis, are enriched by mechanisms that are regulation of cytokine production, apoptotic processes, and proteolysis. So once we confirmed that we could identify proteins associated with cognitive changes in this early phase, and that these proteins are different between amyloid positive and negative, we then, within the preclinical AD group, stratified the analysis by sex. I'm not showing now the volcano plots and the results, but what's really, we were surprised to see is a very small overlap of the significant proteins within the preclinical AD phase between men and women for both platforms. And even when the proteins were shared significant, as you can see in the bottom plot, you have the 12 proteins that are commonly shared in the Olink platform. You see that 3 of them, for instance, are showing an opposite direction of the effect, which means a sex-specific effect. And we saw similar pattern for the SomaScan 29 proteins that are shared, in which 6 of them are showing a sex-specific effect. So this seems to suggest that not only sex-specific signature, but maybe potential sex-specific pathway could be linked to the early cognitive changes in preclinical AD. And when we perform the enrichment analysis, what we see is that in men, proteins that are associated positively with cognitive changes are proteins enriched on nervous system development and neuron differentiation, and mainly synaptic proteins, while proteins that are associated negatively with cognitive changes are enriched by apoptotic processes, regulation of immune response, and response to reactive oxygen species. Is when we repeated the analysis in the women group that we found significant difference in terms of pathways associated with cognition, with proteins that are associated positively with cognitive changes being enriched on extracellular matrix protein, proteins that are present in extracellular exosomes, vesicles, and protein related to growth factor activity. While proteins associated negatively with cognitive changes are proteins that are implied into the regulation of cytokine production and presynaptic assembly. So I think it's worth to note that several proteins replicated in both oligosoma scan with the same effect in men or in women, and I would like to just highlight a few of them. We identified several synaptic proteins or involved in the synaptic plasticity, like cerebellin-4 and stromal cell-derived factor 1, that were both significantly associated with cognitive changes with a positive effect in women, while the matrix metallopeptidase 9, we found it associated negatively with cognitive changes in men. And this is a protein that is involved in extracellular matrix remodeling, blood-brain barrier permeability, and is already reported as associated with cognitive decline at a later stage in MCI and AD, while we also see this effect in preclinical AD. And I would like to conclude saying that proteins associated with early cognitive changes are revealing distinct molecular profile between amyloid positive and negative individuals, pointing to amyloid-specific mechanism before symptoms appears. And that sex-stratified analyses are revealing distinct molecular proteins and molecular processes associated with cognitive changes in men and women. And I would like to add that heterogeneity is not noise that we should correct for, and these stratified analyses are revealing subgroup-specific molecular profiles that are underscoring the urgent need to shift towards a more personalized approach in Alzheimer's disease. And with that, I would like to thank all my colleagues and collaborators from the Barcelona Beta Brain Research Center, as well as the Alpha Plus participant, the Bright Focus Foundation for sponsoring this research, the GMPC for the SomaScan measurement of the Alpha Plus, and all of you for your attention. Thank you.
Okay, if anyone has a question, please step up to the mic. We have about 5 minutes. Yeah, go ahead.
I was wondering, have you been able to replicate this in another cohort, independent cohort, or are you looking to do so?
Yeah, thanks for the question. It's actually ongoing, so we are already collaborating with different groups to try to replicate these results. It's a bit complicated, the fact that the Alpha Plus participants are already pretty unique cohort because they are all cognitively unimpaired, and they are being recruited to be a higher risk of Alzheimer's disease, and finding similar cohorts is quite difficult, but we now have some data for replication, but I still don't have the result to show, but I will be happy to do it next time. Thank you.
Hi. Thanks for a really exciting presentation, Eleanor Drummond, University of Sydney. I was wondering, I could see that you stratified your groups by A beta positive and negative. Did you look at the tau status of the people at all? Was this considered?
Sorry?
Did you also look at whether they were tau positive and negative?
Yeah. Yes, of course. We know that tau is the measure factor for increased cognitive decline. However, we have very few tau-positive individuals in the Alpha Plus cohort, and stratifying also why this group is very difficult. We plan to do some sensitivity analysis, but yeah, still ongoing. Yeah. Thank you.
Go ahead.
Hi, thank you for the talk. I was wondering, given the theme of heterogeneity, if you've if you looked into any clustering of patients or into a method such as the subtype and stage inference or anything like that?
Yeah. We started to work on clustering of protein, like the co-correlation, weighted co-correlation analysis, and we found some clusters that are enriched on neuronal proteins that are associated with cognitive changes. For a matter of time, those results are not in this presentation. But yeah, this is disentangling partially the heterogeneity, but still I think there's a lot of understand.
Thank you.
Thank you.
Hi, nice talk. So my name is Michael Beloy from WashU. I'm wondering, you find sex-specific genes, and then you do pathway enrichment analysis, but different genes may cluster onto the same pathways, right? Even if you identify in different sexes. Have you checked for sex specificity, like carefully in the pathways themselves? Because you might get significance, but your gene ratios might be the same within the different pathways, right?
Definitely. Thanks for the question, because this is a very tricky point. When you do enrichment analysis, it's very complex to understand, because one protein can deactivate another protein, and then the relative abundance can be increased or decreased, but they part of the same pathways. What I was showing here is pathways that are strongly associated with cognitive changes in terms of, I was not showing the pathway that are mostly enriched, like ordered by their significance, but the pathways that have, I didn't explain for a matter of time, but the pathways that have average effect size of the proteins included in the pathways higher. So those highlighted are those that are strongly associated, and when we see differences, we can say that these pathways are strongly related with cognitive changes during time in one group and not in the other group. So this is kind of already a step to disentangle it, but definitely is, if you have an idea to do this better, I would be happy to hear, because yeah, it's a very tricky part.
Thank you.
Thank you.
And this is our last question.
Yes, exciting talk. Mohammed Ali from WashU, and my question is about the differentially expressed proteins. In the SOMA-scan, I saw that you identified more proteins as compared to in the O-link. Is it because of like the platform difference? Maybe SOMA-scan 7K and O-link HT having 7,000 versus 5,500? Or is it like the limitation of the O-link that you don't observe as many proportionally if we look at the total number of gene profile versus how many do you observe to be differentially expressed?
Oh, that's also a nice question. I think it's more related to the fact of SOMA-scan having 7,000 proteins, aptamer measurement, instead of the 3,000 of O-link, to avoid this bias, to do a multiple comparison correction. I was showing nominally significant results. So that's why probably you have more protein significant for SOMA-scan. However, what we are also highlighting the robustness of the protein that we find in common. The common protein between SOMA-scan and O-link are 2,000, which is almost 2/3 of the O-link panel. So in terms of that, I saw that the significance is pretty equal. So I would say it's not a pro and cons, it's just a matter of a bigger panel for SOMA.
Thank you.
Thanks.
All right. Thank you, Frederica.
Thank you.
So move forward to the second talk. It's given by Dr. Keenan Walker. The title,
Hamilton's going to go next.
excuse me. So the next talk will go with the Dr. O. That the title for his talk is Synapse Protein Signatures in CSF and Plasma Predicting Cognitive Maintenance versus Decline in Alzheimer's Disease.
Okay, thank you. Is Mike's good? Okay. Hi, good morning, everybody. My name's Hamilton. I'm a postdoc at the Icahn School of Medicine at Mount Sinai. And today I'll be presenting some work from my PhD and PhD at Stanford with Dr. Tony Weiss-Correa, and also some of this new unpublished work with Dr. Alison Goat and many others. So first to sort of set the stage, we all know, but I want to emphasize that Alzheimer's disease at the end of the day is really a debilitating dementia. And this is sort of beautifully captured by William Ottermohlen's self-portraits. And we all know this, but I think we sometimes get lost focusing on amyloid and tau. These are the hallmark pathologies of Alzheimer's disease. The biomarkers for these are really amazing and have improved the field dramatically. But there's still many holes and missing pieces to the story. And the main question we wanted to get at is, can we identify these sources of heterogeneity in rates of progression and rates of symptom onset in Alzheimer's disease? So we know that even if you have amyloid or tau, who actually ends up with full-blown dementia, it's super heterogeneous, and the rates especially. So you can see there on the graph the CDR sum of boxes score, so that's a dementia severity score across time, across all of these different individuals, and you can see the rates are really drastic. Here's another study, Van der Veer et al. In Neurology 2024. Very similar, basically showing that from people with MCI, the rates of cognitive decline based on the MMSE cognitive score are super heterogeneous. This is mostly based on people who are amyloid positive, but actually even tau, so we normally think of tau as sort of this late stage marker that tracks with symptoms, but even tau, it's also sort of really quite heterogeneous. So this is the ROSMAP cohort where we have neuropathology measurements. So we have, everyone here is amyloid positive, and you can see the global cognition, so lower is worse, versus their tau tangle load. And so it is true that if you have tons of tau, you probably have dementia, but actually early on, if you have sort of low, mild levels of tau, there's still a really wide spread of who's cognitively normal versus who has full-blown dementia. And so we thought that maybe the cerebrospinal fluid might hold the answer to explain who is it among those people who have amyloid and tau, who is it that actually has dementia and who's cognitively resilient. And so we had a really big collaborative study across many institutions. This was my PhD work with Dr. Tony Weiss-Correa, and we collaborated with groups from WashU with Carlos Kutraga and also from Finland with Ville Leinonen. And so we had 5 cohorts, a mix of sporadic disease, sporadic Alzheimer's, autosomal Alzheimer's, and 5 cohorts, roughly 3,400 subjects. And the key here was that we actually had basically integrating multiple layers of data. So we had not just the CSF proteomics and the cognitive scores, but we also had amyloid biomarkers on basically everybody. And so we can ask, what are the proteins that can separate people who have similar amounts of amyloid and tau, but then have very differing levels of their cognition? And so we tried to first ask this question, what are the protein correlates of cognitive impairment independent of Alzheimer's pathology? And so what we discovered from this unbiased search of all these proteins, trying to identify which change with cognitive impairment independent of CSF p-tau A-beta ratio, we found sort of this distribution where actually a lot of these are synaptic proteins, but MPTX2 was the most downregulated protein with cognitive impairment. YWHAG, or 1433 gamma, was the most upregulated protein. And if you look at pathway analysis across all the proteins, it's highly enriched for synaptic proteins. And so perhaps we're actually, it's known that synapse loss is one of the most strong, at least histological correlates of cognitive impairment. Maybe we're capturing some of this from the CSF. We next tried to identify a multi-protein signature that could capture the changes in all of these synaptic proteins with cognitive impairment. And so we used just a basic linear model, machine learning model, to try and predict cognitive impairment based on all of those hundreds of synaptic proteins. And what we found was that the model actually only needs 2 proteins. So it just needs the top and upregulated and downregulated proteins, YWHAG and MPTX2. What's really interesting, I think, is that the model spit out basically a one-to-one relationship between these 2 proteins, which everything was log normalized here. So this is basically a ratio between these 2 proteins as our sort of synaptic signature of cognitive impairment. And so we looked at this ratio across all of these cohorts, and we saw it was really quite strongly correlated with the dementia severity, even separating out cognitively normal versus MCI, and then moderate to mild to moderate severe dementia. This was also the case for the Diane cohort, which all the way on the right, which is this autosomal dominant Alzheimer's disease cohort. So it's true also for this more pure AD. The key here was looking at this ratio and its relationship with cognitive impairment independent of the pathology, right? And so we have on the y-axis the ratio, the synaptic ratio. On the x-axis, we have the p-tau A-beta, so how much sort of amyloid and early tau pathology you have. And you can see even among the people who are all amyloid and tau, early tau positive, the ratio is really what separates out who actually has full-blown dementia versus is cognitively normal. We think these people might be resilient, or at least some of them. What's also interesting is that this is not just a marker that correlates with dementia severity, but also starts changing really early on with just normal aging. So this is looking at cognitively normal people across all of these cohorts, basically all the controls. No one here has Alzheimer's mutations, and we found that the synaptic ratio actually changes really quite early on, basically like as early as we measured. Around age 20, it starts, there's a increase in the slope, whereas p-tau A beta actually is relatively flat, basically your whole life until at some point you start getting amyloid. And so we think that this might be an early indicator of the synaptic aging process. It's pretty interesting. There's some studies in non-human primates showing that changes in synaptic morphology and synapse loss is actually a really early indicator of just the normal aging process, and that it's highly correlated with cognitive function in the non-human primates. So maybe we're capturing some of this. What's really striking is that if you actually look at the people with the AD mutations, those are the AD mutation carriers versus the non-carriers, they actually show faster increases in this ratio over time. Right, so these are the carriers in the red, non-carriers in the gray, suggesting that potentially this Alzheimer's might be an accelerated form of synaptic aging. And we actually showed that if you look at the synaptic ratio across estimated years until symptom onset, so this is basically based on which genetic Alzheimer's mutation you have, you can sort of estimate when someone is going to get symptoms. And so we're plotting the ratio across this axis, and you can see that it starts, the carriers start to separate around 20 years before estimated symptom onset compared to the non-carriers, showing that it really is an early indicator, and that really actually dramatically increases in slope with progression. So we're capturing both stages of the disease, not just the late stage, but also early stage. The key was actually showing that it predicted actual changes in symptom onset. So before it was estimated symptom onset, now we're looking at not autosomal dominant AD, but in the normal population across all of these cohorts. We have a baseline CSF proteomics measurement, also baseline biomarkers, and then 15 years of longitudinal cognitive follow-up. And so we can ask, does the ratio at baseline actually predict who's going to decline in the future? And so we first looked at people who are amyloid and tau positive, or early tau positive, and had MCI at baseline, mild cognitive impairment. We also stratified the synaptic ratio into these 5 different groups, basically, low to very high. And you can see that it really sort of remarkably stratifies who declines and when. And so actually, if you have really high levels, basically everyone declined almost within a year, and really low levels remained cognitively, or at least they didn't experience more cognitive decline over the 10-year follow-up. These were really quite low sample sizes, but I think it's pretty interesting. And even actually, if you look at sort of the in-between levels, you can see this sort of stepwise increase in the rates of cognitive decline. And so I think this biomarker could be used not just for defining your positive or negative, but having different stratifications. What's I think also important is that we also tested this in the cognitively normal people at baseline. So if you're cognitively normal, have early signs of amyloid and tau, can this biomarker predict who gets Alzheimer's, dementia, or mild cognitive impairment at least? And here again, we showed that there's a stepwise increase based on these groups. So it really is both a sort of middle, late stage, and early stage biomarker. Next, I want to talk about some new unpublished work. So we've been working with Rowan Saloner and Adam Boxer from UCSF on now sort of investigating this biomarker in frontotemporal dementia. There were some early papers and recent papers showing that these 2 proteins were the top dysregulated proteins at the individual level, and so we were curious whether the ratio might also be relevant. But actually, first off, this is in the UCSF sporadic Alzheimer's disease cohort looking at the CSF ratio with tau PET. And actually, the synaptic ratio is more strongly correlated with the dementia severity than tau PET is correlated with dementia severity. So again, this is sort of, or maybe capturing synapse loss, synapse dysfunction that's really tightly correlated with cognitive function. It's also interesting that this ratio almost fully mediates the association between tau PET and dementia severity. And so, perhaps the tau pathology is actually affecting directly the synaptic pathway, and the synaptic pathway is also captured in people who are tau negative. In FTD, it's elevated across sporadic and familial FTD. So this is looking at cognitively unimpaired versus sporadic FTD, sporadic AD, and then looking at the familial FTD, so people with the mutations. There's 3 different types of mutations in FTD. There's the C9, ORF72, GRN, and MAPT. And basically, they're all super elevated at the symptomatic stage, but actually all statistically significantly elevated in the pre-symptomatic stage as well. And I think what's really key here is that we could actually predict, even in familial FTD among pre-symptomatic carriers, who actually ends up with dementia in the near term. So this is people looking at the synaptic ratio versus plasma neurofilament. And you can see in the red, these are all the people that declined over the follow-up of 8 years. And it's people who are double positive that really decline. You can see this dramatically here, and actually also this is looking at the rates of the change of the CDR sum of boxes dementia score. And really, it's quite obvious that the double positive group is really at risk. And you can see actually if you're positive for neurofilament but negative for this ratio, you're actually relatively safe over the short term, at least. And so we think that this could be a useful sort of staging system, at least for frontotemporal dementia, of looking at doing annual monitoring of plasma neurofilament, and then once you become positive for neurofilament, then maybe check the CSF to see if you're actually likely to decline. So in summary, we found that the CSF YWHAG-MPTX2 ratio is one of the strongest known molecular correlates, biomarker correlates of dementia severity in AD and FTD. And importantly, it also predicts future AD and FTD symptom onset among people who are cognitively normal and are at high risk. So it predicts long-term cognitive maintenance versus decline among high-risk individuals. Some future directions and implications. I think it suggests that synaptic dysfunction is really likely an early, maybe one of the earliest pathological events in Alzheimer's disease, maybe even before that with aging, and not just a late-stage pathology, and that this ratio has really promising utility in Alzheimer's and FTD clinical trials. For patient stratification, potentially as an endpoint, and potentially to identify responders and non-responders to certain drugs. And lastly, I'd like to thank a ton of people were involved in this work, but I'd like to especially thank my advisor, PhD advisor, Tony Weiss-Correa, crucial collaborators, Carlos Cutraga, Rowan Saloner, Adam Boxer, and my postdoc advisors, Allison Goat and Scott Russo. Thank you.
Thank you, Hamilton. We're going to have to skip questions, but if we have time at the end, definitely feel free to come up and ask him a question. Yeah, go ahead. Have a seat.
All right, let's welcome the third speaker, Dr. Keenan Walker. The title for his talk is Plasma Protein-Wide Analysis of Dementia Risk Mechanistically Implicate Synaptic Biomarkers.
Okay, thank you, Teresa. So Hamilton nicely gave an introduction to the importance of synaptic biomarkers. And so in contrast to the CSF approach which he took, I'm going to talk about our pursuit of synaptic biomarkers for dementia in blood. All right, so we know Alzheimer's disease is defined by amyloid and tau, but in addition to that, synaptic dysfunction is a core feature. And this includes things like presynaptic degeneration, synaptic clearance through microglial removal, astrocytic removal, and A-beta-mediated toxicity on the synaptic cleft. And we also know that Alzheimer's disease, starts very early, decades before the onset of symptoms, decades before the onset of dementia. The consensus now is that Alzheimer's disease pathology starts as early as middle adulthood. So we wanted to identify an early biomarker for synaptic dysfunction in blood that would be predictive of dementia risk in subsequent decades. So we went about doing a 25-year proteome-wide analysis of incident dementia risk. And I'm focusing here on our examination of synaptic biomarkers and the subsequent analysis that followed. So these are initially results from a paper that was published 2 years ago in Science Translational Medicine, where we did a proteome-wide analysis of dementia risk in the atherosclerosis risk and community study. So this is a cohort of almost 11,000 people. We had 5,000 proteins measured with the SomaScan platform, and we looked at each of those proteins in relation to the dementia risk over the subsequent 25 years. So here was our first set of results where we have proteins in green that pass the Bonferroni corrected threshold for multiple comparisons after adjusting for demographic characteristics, cardiovascular risk factors, and other relevant confounders. So we took these 26 and then we combined them with proteins that were associated with dementia risk, importantly when measured in midlife over the next 15 years. So we looked at a shorter follow-up window and found actually a separate set of proteins, mostly non-overlapping with our initial set. So all in all, we had 32 proteins that were associated with dementia risk when measured in middle-aged adults. And I highlight here the ones involved in synaptic function. Well, I bolded them, but I want to also make it clear that there's plenty of other biologic pathways that are implicated, including immune function, proteostasis, vascular, and metabolic function as well. So we then took data that was previously published by Johnson and Wingo and colleagues, and we looked at whether or not the proteins that we measured in blood that seemed to be relevant at midlife were also differentially expressed in brain tissue of individuals with Alzheimer's disease at autopsy. And the finding that really stuck out to me was this idea that a lot of the synaptic proteins were actually increased in blood, but downregulated in brain tissue, at autopsy. And we saw this pattern over and over again. It wasn't, it didn't happen all the time, but it was fairly consistent. So, that got us interested in whether or not, it made it clear that these synaptic proteins seem to be relevant, late in the disease, but also there's some signal early in blood, as early as midlife. And then we set out to understand if, perhaps there's this causal link between the synaptic proteins and dementia risk. And we did this the best we could with observational data using two-sample Mendelian randomization. So we ran a GWAS on the blood levels of our synaptic proteins, found protein quantitative trait loci, or PQTLs, for those synaptic proteins, and then related them to Alzheimer's disease using this two-sample Mendelian randomization framework. We also looked in a reverse direction to see if Alzheimer's disease was indeed causing a differential expression in these synaptic proteins in blood. And in the forward direction, relating proteins to Alzheimer's disease, we found one protein, calcitonin 3, that whose greater levels in blood was purportedly causally linked to reduce Alzheimer's disease risk. But most of the action was in the opposite direction, where we looked at Alzheimer's disease as a driver of proteins. And we found, out of the 8 synaptic proteins we were interested in, 4 of them seemed to be differentially expressed in blood as a result of Alzheimer's disease. And this sort of genetically validated these proteins as potential biomarkers, but also in the context of our analysis, we're seeing these proteins differentially expressed in the 4th, 5th, and 6th decade of life. So we went on to look at our synaptic proteins, where we think they actually might be mechanistically relevant in brain tissue. And so we performed a similar approach where we took genome-wide association data that was generously published by Dr. Thomas Wingo's group, and we took those genetic instruments for our synaptic proteins and using the two-sample MR framework related them to Alzheimer's disease. And of the 8 proteins, we had genetic instruments for one of them, complexin 1, and we actually found in this two-sample MR framework that greater levels of complexin 1 were associated with increased Alzheimer's disease. And importantly, this is greater levels of complexin 1 in brain tissue. We didn't find genetic instruments in plasma, so we couldn't test that effect, but we did see something for brain tissue. So we looked into this further, complexin-1, and we see that's clearly a synaptic protein. If you look at the Go terms, over 90% are annotated as synaptic. The other 10 is involved in endolysosomal function. We looked at the network of complexin-1 proteins using protein-protein interaction, string pathway analysis, and we see a lot complexin-1 strongly connected with a lot of proteins, a lot of synaptic proteins that we are already aware of in the ADRD space, including things like SNAP25, the VAMPs, and syntaxin. So, clearly relevant here to some of the things that we're very aware of as potential biomarkers. But when we functionally annotate the network of proteins sort of related to complexin-1, we see it's involved in neurotransmitter secretion, synaptic vesicle exocytosis, and particularly relevant is this snare complex assembly. So we look further into this, and we see that complexin-1 is this presynaptic protein really involved in vesicle exocytosis, so the release of neurotransmitters into the synaptic cleft via the snare complex here. So, it's sort of involved in this machinery. So we wanted to see, now we got this, putative biomarker complexin-1 in blood that we think relates to the synaptic process, particularly breakdown in presynaptic signaling. But we want to see what high levels of complexin-1 in blood meant neurobiologically, from a CSF perspective. So we were able to obtain blood and CSF from, in the same participant from 200 people in the Johns Hopkins CSF cohort. And we related plasma levels of complexin-1 to CSF biomarkers as measured by the new ELISA CNS 120 panel. And here are the results from, this volcano plot, just adjusted for age and sex. But we see people with high levels of complexin-1 in blood have CSF levels of some of the biomarkers for Alzheimer's disease that we're very aware of, like p-tau 217, 231, and 181. And if we zero in on just the amyloid and tau biomarkers here, I think it makes the picture a bit clearer. Now, we hypothesized a priori here that our complexin-1 would be most strongly associated with synaptic biomarkers in CSF. And, this wasn't fully the case. We did see some relationships didn't pass multiple correction for multiple comparisons, but the biomarker that sort of was at the top of the list was SNAP25, which is also involved in the snare complex, sort of supporting the role of complexin-1 as an indicator of this presynaptic process. We also looked at how complexin-1 in blood related to CSF immune proteins and found that IL-12, which has previously been implicated in AD in CSF, was sort of at the top of the list, plus a number of others, including IL-9. So, to gain further information about the role of this protein, what it means to have high levels of complexin-1 in blood, we ran complexin-1 in blood against the full CSF proteome. So we measured 7,000 proteins in CSF using the SomaScan platform, and we related complexin-1 in blood to all 7,000 of these CSF proteins. This volcano plot nicely shows the results of that analysis, but they were much more interpretable when we sent them to pathway analysis. And what we found was, in terms of the upregulated proteins here, higher levels of complexin-1 was associated with a number of proteins in CSF that were associated with immune disease, namely lupus, infection, and then processes such as focal adhesion and coagulation. So this is sort of more of an immune than a synaptic picture, and this was somewhat surprising. When we looked at these proteins here on the sort of other side, these are proteins downregulated in CSF in people with high complexin-1 in blood. We found, again, immune processes, but they were sort of, these are downregulated immune processes, including antigen presentation and cytokine response. So looking at complexin-1 across the age range, this is complexin-1 measured in blood in a Baltimore longitudinal study of aging, where we're lucky enough to have measurements from age 20 to age 100. We see this sort of inflection point around age 60. And when we stratify by people who subsequently develop dementia, we see this sort of uptick in around midlife in complexin-1 in blood in people who go on to develop dementia. And this uptick comes as early as midlife. Let's turn turn that off. All right, so just coming full circle here, we do see, and this is sort of what got us down this path initially, complexin 1 in midlife associated with long-term dementia risk, and it seems like it's this highest tertile. So this is this third tertile associated with greater dementia risk over this 25-year period, and this is sort of demographically adjusted Kaplan-Meier here. But when we look at complexin 1 in our 75-year-olds in late life, we see an even stronger effect as we might expect. But again, it's people with the highest level of this protein. So with that, I'll end. Just in summary, brain complexin 1 was causally implicated in Alzheimer's disease. It is elevated in midlife in blood, well ahead of dementia onset, and we found that high levels of blood complexin 1 to be associated with a number of those p-taus in CSF, SNAP25, and a number of inflammatory proteins. And surprisingly, high levels of blood complexin 1 seem to be linked more so to immune processes in CSF. So I think our results suggest this crosstalk between presynaptic dysfunction and neuroimmunity. And some of the implications might be synaptic dysfunction giving off damage-associated molecular patterns and activating the neuroimmune process, or it may be an indication of synaptic sort of removal, which is a glial or immune-related process, and we're seeing that signature in CSF perhaps. And with that, I'll end. Thank you for your attention, and I want to thank those who helped with this project, particularly members of my lab, especially Cassie Blue, who ran a lot of the analysis for this project. Thank you.
In the interest of time, we may have 1 or 2 questions.
All right.
All right.
Save some time. Okay, so it's my pleasure to introduce Dr. Teresa Tian. Dr. Tian's gonna present her work on the proteomic signature of dual cognitive and mobility decline in older adults.
So I'll be talking about proteomic signatures of dual cognitive and mobility decline. This is a model of tracking pathophysiological changes in the development of Alzheimer's dementia. So it begins with the dynamic change in beta amyloid, followed by the tau and brain structural change, and subsequently with the deficits in memory and clinical function. There's one important component that is not considered in the picture is mobility impairment, such as slow gait. Older individuals with mobility impairment are twice likely to develop dementia compared to those who do not have mobility impairment. And this change could occur later in the stage, could also occur early in the stage during the normal aging process. So in the general aging population, we can picture several phenotypes. One phenotype is a parallel decline in both memory and gait. There are also individuals who decline only in the memory or only in the gait. And this can be operationalized by assessing their memory performance and gait speed over time. It is striking that when we look at the association with the future dementia, individuals with a dual decline in memory and gait speed had substantially higher dementia risk compared to the individuals who decline in memory only. And this finding has been replicated worldwide across several aging cohorts. And I wanted to note that this finding has been fairly consistent, regardless different cutoffs to define the decline in memory and gait, and also consistent regardless of different memory tests used in each study, and also using either self-report or performance-based gait speed. So over the past decade, there has been studies looking to study this phenotype in clinical samples and aging population. But a more interesting question is, what are the underlying mechanisms? So do individuals had a dual decline show substantially different biological processes compared to others? Whether this implicate a specific pathway to dementia? So why there's a link with the high risk of dementia? And eventually, if we can identify these biomarkers, we can use them to identify people at risk for future dementia, and also, if possible, to design a strategy for early prevention. And this fits into the geroscience hypothesis that to understand the underlying biological processes, it can capture the conditions that has a clear definition as disease. But also, in this aging process, there are conditions that are not quite clear as a disease, but it happens to most of aging individuals. So aging is what occurs to all individuals, but the disease only occurred to a proportion of them. It has been hypothesized that the dual decline have several different causes. One could coming from the central nervous system, but also could be coming from more systematic causes, such as metabolic dysfunction, impaired lipid metabolism, or vascular burden. In this study, we aim to identify the plasma proteomic signatures of dual decline, as well as gait decline and memory decline, by comparing them to the no decline group, which is considered as the reference group. We use the data from the Baltimore longitudinal study for aging, so the phenotypic change in memory and gait, as well as the plasma proteomics, are assessed within the same time frame longitudinally. So the data are collected between 2006 and 2020. Here is the plasma SomaScan 7K platform. Here's the participants' characteristics. Average, the participants are in their 70s, and I want to note that at baseline, the dual decline do not differ in memory or gait compared to the no decline group. And we use the linear mixed effects model to compare the differences at baseline as well as longitudinal change over time. Interestingly, there's no significant baseline differences in any of the proteins between the declining group and the reference group. But it's striking that over time, only the dual decline group showed significant alterations in certain proteins compared to the no difference group. And there's no differences between the gait or memory decline only group with the no difference group. So we further look into the biological domains of these plasma proteomic signatures of the dual decline. This is a fairly busy figure, but the top-ranked biological domains include structural stabilization, immune response, proteostasis, synapse, and lipid metabolism. Here I'm highlighting the top significant proteins. PGP 9.5 was the top significant protein that significantly decreased in the dual decline group. It has been involved in immune responses, inflammation, interaction with the mitochondrial proteins, and oxidative stress. Similarly, activin A and laline are also important for mitochondrial function. The TREM2 has been involved in microglia activation. TRIM72 has been involved in the membrane repair triggered by mitochondrial dysfunction. And here we also see the complexin 1, which is the signature synaptic protein. So we further validate these protein signatures with future cognitive impairment, and large proportion of the proteins are also significantly associated with the impairment. And they're also in a consistent direction. That means the protein that significantly increased in the dual decline group, they also associate with the increased risk of dementia. We also further validate with the change in brain atrophy based on the imaging brain MRI and also the blood biomarkers of Alzheimer's disease. We can see select proteins also associated with these biomarkers, especially in the medial temporal lobe and p-tau 181. We further performed pathway analysis using the gene set enrichment analysis. The dual decline showed 116 significantly enriched pathways. And it is the top significant pathway is mitochondria protein degradation, followed by the degradation of the extracellular matrix. And there are 21 pathways related to memory decline. There are 20 pathways related to gait decline. Except the post-translational modification pathway, all the pathways in the dual decline are unique. They're not shared with the memory or gait decline, which may suggest a specific pathway to dementia. We further performed IPA analysis, which provide complementary information. The top significant pathways are involved with the neurotransmitter release cycle and also the protein phosphorylation. And some of the pathways are also significantly associated with future impairment, brain atrophy, and blood biomarkers. So the take-home message, the key findings from the study is that the individuals with a dual decline within aging, they showed significant changes in certain protein signatures. And by looking at the signatures, they may indicate mitochondrial dysfunction, proteostasis, neuroinflammation, and immune responses. So here is a paradigm of aging hallmarks, which coming from this study, the dual decline has been related to 3 of the aging hallmarks here, including inflammation, mitochondrial dysfunction, and the loss of proteostasis. So here's a quick snapshot of the studies to date. There has been a lot of study going on looking at the underlying mechanism between the dual decline and dementia. So these studies looking at the clinical characteristics of the dual decline, as well as the neuroimaging profiles. And there's a lot more omic studies going on to further elucidate the mechanism. The study has been recently accepted for publication, should be out very soon. I'd like to acknowledge the team and also the NIA Intramural Research Program. Thank you.
Thank you, Teresa. So we have time for a few questions. Okay, so I have one. The proteins that were implicated in dual decline, and do you have a sense of their origin? I know, thanks to Hamilton, et cetera, there's been a lot of interest in where proteins are coming from that are associated with various phenotypes. Now, did you see more proteins from like muscle versus brain or some other tissue, or do they tend to be nonspecific?
I think it's pretend to be non-specific, and we do see there are proteins related to the synaptics and also the skeletal muscle mitochondrial function. So I think it's picturing something more systematic, which could also relate to the crosstalk between the brain and the muscle, or the brain and the lower extremity.
Thank you. Okay. Well, thank you, Teresa. Okay, I'll go ahead and introduce the next speaker. It's my pleasure to introduce Dr. Liu. He's going to talk about plasma proteomics and how they reveal novel protein signatures associated with dementia and chronic pain.
Yes, good morning, esteemed colleagues. My name is Liu Xiaoduo from Xuanwu Hospital, Capital Medical University. And it's a privilege to present our work. Plasma proteomics reveals novel protein signatures associated with dementia in chronic pain individuals, a prospective cohort study. And to begin with, I want to make a few points, and the photo videos and the audio recordings are prohibited in this presentation. And yes, and there is a research background. And as we all know, cognitive impairments and dementia, especially Alzheimer's disease, pose an immense and growing global burden, and simultaneously, and chronic pain affects over 1.6 billion adults worldwide, with prevalence rising sharply with age. And crucially, and robust evidence links CP to increased dementia risk shows that a large analysis, including 35 cohorts, and found CP increases all-cause dementia risk by %24, and AD risks by %28. And the UK Biobank specifically showed multi-site CP increased dementia risk by %36, and demonstrating a dose-response effect. And the CP patients show higher rates of neuropsychiatric symptoms and reduced cortic sickness. And mechanistically, and persistent pine signals engage pathway overlapping with AD pathogenesis, and including neuroinflammation and locus coeruleus and norepinephrine system dysfunction. And just for the necessity and illustrating the CP2-AD link is vital for early identification and target intervention and precision prevention in this large vulnerable population. And we lack validated biomarkers for AD risk predictions specifically with CP cohorts. And for feasibility and high throughput plasma proteomics like Olink and offers unprecedented and sensitive to profile thousands of proteins simultaneously. And I will give you a brief introduction to the methods we used. And first of all, and for the study population and over 1,500,000 participants with baseline plasma samples. And for chronic pain definition, and is a self-reported pain in over one size, including head, face, neck, shoulder, back or stomach, and knee, and west bright, which is lasting over 3 months. And for the outcomes and incidence all-cause dementia and Alzheimer's dementia and vascular dementia identified it through hospital records over the follow-up. And for the proteomics, and we analyzed 2,923 plasma proteins using 4 Olinks panels, including oncology, neurology, inflammation, and cardiometabolic. Yes, and for the analysis, and first we use the Cox models to assess longitude association between each protein and dementia risk. And then we use the function enrichment, KGG, and GO to identify key biological pathway. And third, we use the C index to rank proteins by predict power. And after that, we evaluate top 5 proteins with highest C index values using AUC analysis. And the Kaplan-Meier curves compare high versus low protein expression. And last, we explode the associations between top proteins and structural MRI measures. Okay, next comes the analysis of the results. And this figure illustrates the longitudinal association between plasma proteins and the risk of dementia. And we can see that GFAP and GDF15 showed strong associations across all 3 dementia subtypes. And NEFL was particularly relevant for Alzheimer's disease. And in contrast, proteins like HPGDS and ITGAV were negatively associated with dementia risk. And to understand the biological relevance of dementia-associated proteins, and we performed GO and KGG enrichment analysis. And for all-cause dementia, and the enriched terms included cytokine, signaling, and cell adherent, and extracellular pathways. And in contrast, proteins linked to Alzheimer's and vascular dementias were mainly involved in the neural process, such as astrocyte projection and synaptic transmission. Okay, here we present the top ranking proteins based on their C-index values for predicting dementia outcomes. And NEFL and GFAP and GDF15 constantly appeared among the top predictors of across dementia, and their high C index values above over 0.75, and highlight their robust discriminative power. And to evaluate the predict utility, and we calculated the AUC for the top 5 proteins across dementia subtypes, and NEFL, GFAP, and GDF15 constantly showed good performance. And across all 3 dementia subtypes, and higher level of NEFL, GFAP, and GDF15 were constantly associated with significantly lower dementia-free survival over time. And this figure shows association between key proteins and the brain structural volumes. And NEFL and GFAP were linked to widespread cortic atrophy, particularly in frontal and temporal regions. GDF15 was associated with both corticosteroids and reduced subcortic volumes. And having reviewed the key findings, and we now move on to the discussion. And in summary, our proteome-wide analysis identified several proteins, particularly NEFL, GFAP, and GDF15, that were constantly associated with increased risk of dementia across subtypes, and functional enrichment supported links to neuroinflammation and synaptic pathway. And to conclude, with our study offer promising insights into proteomic biomarkers of dementia risks among individuals with chronic pain, and it remains a preliminary step. With further refinement, plasma proteomics could pave the way for personal risk subgroup analysis and target prevention in chronic pain population. And before I closing, and I'd like to sincerely thank Professor Tang Yi for his guidance and leadership, as well as all the members of the Tang Lab for their valuable contribution and to this work. I also wish to acknowledge my first co-author, Dr. Gao Peiyang, and our collaborator at Xuanwu Hospital. And finally, we are deeply grateful to the UK Biobank participants and staff. And thank you for your attention.
Okay, we have a few minutes for questions. While we wait, I'll ask one. So, number of proteins you identified among the chronic pain individuals. Do you have a sense that the proteins associated with dementia among this group, differ, in a meaningful way than the proteins associated with dementia in the broader population?
And we just analysis this protein in chronic pain, but we and I think we will do it in the future research to answer in the broad dimension.
All right, thank you.
Thank you.
Another question.
Yeah, Andrew Tobin from the University of Glasgow. Thank you to you all for really inspiring talks. I guess this is a question for you, for the panel as a whole, really. Are you seeing an emerging picture of adaptation when you look at mechanisms? Are you able to dissect out what is disease causing, what is disease marking, and what is adaptive changes to disease? And a follow-up question to that, which is sort of linked, is are your biomarkers, when you're looking at the plasma, now very associated with the permeability of the blood-brain barrier, and how much does that implicate? So I guess there's two questions in there.
Okay, and I think first of all, and we use plasma level and in the UK Biobank, and we didn't try it in the brain tissue or the CSF tissue, and I think we will continue to use such as different data sets to evaluate it and the mechanism in the BBB and such as the nerve system in the brain tissue. Yeah, yes.
And so I can shed some light on the first part of your question. So I've been really interested in what we see when we test for the causal link versus what just we see in associative analysis. Oftentimes we see reverse associations, where with Mendelian randomization, you'll see a positive association, but then when we look at the relationship, just with a normal, survival model, we'll see the opposite. What's been interesting is when we look at proteins associated with dementia risk across different time periods, when we measure proteins in blood, and I'm speaking only in blood, in midlife, they tend to be more consistent with the Mendelian randomization effects than when we measure the same proteins in late life in terms of their relationship to subsequent dementia risk. So I think what we're seeing in midlife is more, and this makes sense, again, given the how long we think these proteins are like playing a role in the disease mechanism, but the midlife proteins and that relationship seems to be more consistent with the causal association, if they are indeed causal, versus late life. I think we're seeing either a response, so we're seeing an increase maybe in protective proteins in response to pathology, and so like TREM2 looks, for example, is high in late life in people at risk for dementia, but we know soluble TREM2 is protective, and so when you look at soluble TREM2 in midlife, you see people with low soluble TREM2 being at greater risk for dementia. So we see this. I think the timing does matter. And to your second question, we are looking at the degree to which proteins, the degree to which they correspond across blood to CSF, and if that matters in terms of their link to dementia risk. And I'll say that a lot of the proteins that are strongly associated with dementia risk either don't show a strong correlation with themselves in CSF, so don't show a strong correspondence across biofluids, or just not expressed in the brain at all and not found in the brain. It's been really fascinating seeing that.
I'd like to just comment to the first point. I think it's a brilliant point that, without the observational studies, we don't know whether it is a response to an insult or it's a causal pathway. So I think to add to Keenan's point that maybe one way to understand the fundamental changes in these proteins in the aging process is whether there's dynamic change early on in life versus a relatively slow down change in later life. So I think this could provide some insight into where this faster rates of change are happening across the lifespan.
And just to add, if I could, if there's nobody else asking a question, are you at a stage now with the antibody treatments and all the rest of it? Are you going now and looking at the proteomic changes associated with treatments that you feel are changing, at least whether it's changing disease or not? Of course, is very very debatable, but it's certainly changing the pathology in terms of that. Are we at that stage where you're able to do those studies?
Yeah, I assume the companies that have these disease-modifying therapies are looking at it. I would love, if they want to send me samples, I would love to look at, address this question.
Key, doesn't it? Because you're measuring the changes, and that's associated with disease, which is incredibly important. And so, but now you've got a little key that's unlocking the key differences, which could be important in terms of, because you're seeing a lot of differences up and down. Like, I think that's.
Yeah, I get the sense they're holding on to the samples pretty closely, but I would love to get my hands on them. Okay.
Okay, thank you.
Pharma's probably doing it. Yeah.
Okay, let's welcome the last speaker. Is Dr. Shrestha. Her talk is Blood Biomarkers of Neuronal Injury and Glial Cell Dysfunction and 25-Year Cognitive Decline and Incident Dementia in ERIC Study.
Hi, everyone. Okay. Well, all right. So I have nothing to disclose. So research on blood-based biomarkers of dementia has made significant advancements over the past decade, but prior research has mainly focused on Alzheimer's-specific pathologies, including amyloid plaques, and the neurofibrillary tangles. As other speakers pointed out, Alzheimer's disease often co-occurs with other non-Alzheimer's disease brain pathologies, and dementia pathologies in general are driven by diverse non-specific processes, for example, astroglial and microglial dysfunction and related neuroinflammation, impaired protein clearance, etc. So a better understanding of comprehensive pathophysiological processes that contribute to dementia is important for better disease risk characterization, diagnosis, as well as determining the therapeutic options. So however, the current understanding of blood-based biomarkers reflecting other pathophysiological processes is limited. And also in general, there are a few prospective studies on examining blood-based biomarkers in relation to cognitive decline and dementia in community-based settings. Prior studies mostly recruited participants from memory-based clinics, and also further prior studies focused on populations of European descent, Black African Americans who are disproportionately affected by the disease are not well represented in this research. So to address these research gaps, we wanted to examine the association of serum biomarkers reflecting some of the non-specific disease processes, including glial cell and neuronal dysfunction with future cognitive decline and dementia in Black and white community-dwelling adults. So in particular, we examined 6 biomarkers, and which included GFAP and YKL, which are associated with astrocyte reactivity, SCD14, which has been implicated in microglia-mediated neuroinflammation in the brain, NFL and total tau, which are markers of neurodegeneration, and UCHL1, which is an enzyme involved in the ubiquitin proteasome pathway that regulates misfolded protein, and it is also considered a marker of neuronal injury. So among the 6 biomarkers, there is more robust evidence on GFAP and NFL, but other prospective evidence on other markers, including YKL40, SCD14, and UCHL1, are very limited. So we conducted this study in the atherosclerosis risk in communities, or the AAIC study. We heard about this study briefly from our prior speaker, but just to reiterate, it is an ongoing prospective study of community-dwelling adults from 4 communities in the United States. Between 1987 and 1989, about 15,700 primarily Black and white adults aged 45 to 64 years enroll in the study, and these participants are followed up periodically through in-person clinical examinations. For the current investigation, we selected a subset of participants who attended Visit 3 exam, in-person exam between 1993 and 1995. So 4 biomarkers, GFAP, NFL, total tau, and UCHL1, were measured in serum samples collected from 1,766 participants who attended brain MRI examination. And these biomarkers were quantified using Samoa by Quanterix. And SCD14 and YKL40 were measured in about 3,000 participants. And these biomarkers were measured as a part of the MARC-VCID consortium. And these participants were followed from Visit 3 to Visit 9 for cognitive outcomes, and the maximum follow-up time was 29.5 years. So our outcomes were global cognitive G-scores and all-cause dementia. So global cognitive G-scores were obtained from 3 cognitive tests measuring executive function, memory, and language, and these tests were administered up to 8 times from Visit 3 to Visit 9. And all-cause dementia was ascertained using in-person cognitive data, using established protocol, and these dementia cases were adjudicated by an expert team of clinicians and neuropsychologists. And in the absence of in-person cognitive data, dementia cases were identified using telephone interviews, informant interviews, and hospitalization and death records. For our statistical analysis, we use shared parameter models to jointly fit linear mixed models and survival models to estimate cognitive decline and dementia rate for each biomarker separately. And this modeling approach accounts for missing data, including those associated with cohort attrition and death. We adjusted for sociodemographic factors, cardiovascular risk factors, and ApoE4 status. So the age of the participants selected for this study ranged from 50 to 73 years. The median age was 60 years. And this figure shows the characteristics of participant at Visit 3 by future dementia status. And those who developed dementia, as indicated by orange bar graphs, were likely to be older, women of Black race, and ApoE4 carriers. So this figure shows the associations of the biomarkers with cognitive decline in the overall sample. So on the y-axis, we have biomarkers, and on the x-axis, we have 25-year cognitive decline associated with 1 standard deviation higher log biomarker level. And negative values imply faster cognitive decline. So as you can see here, higher levels of all biomarkers were significantly associated with faster cognitive decline, and the associations were in particular stronger for NFL GFAP, followed by YKL-40. And this figure on the right shows associations with incident dementia, and on the x-axis we have hazard ratio. Higher hazard ratio implies elevated dementia rate. And higher levels of all biomarkers were associated with higher dementia rate, although the associations were statistically significant only for NFL GFAP, YKL-40, and SCD-14. And as with cognitive decline, the stronger associations were seen for NFL and GFAP. For example, 1 standard deviation higher NFL and GFAP were associated with 73% and 63% hazards of dementia, respectively. And this this slide shows the associations of biomarkers with cognitive decline and dementia stratified by race. And the layout of the slide is similar to the one that I showed you earlier. And the associations for white participants are shown in blue, and the associations for black participants are shown in orange. And the associations are similar in both black and white participants. And we found that these 3 biomarkers, NFL GFAP and YKL-40, were significantly associated with faster cognitive decline and elevated dementia rate in both black and white participants. As for other biomarkers, the associations were not as robust. So overall, we found robust associations for GFAP, YKL-40, and NFL, and these findings suggest early involvement of astrocyte-mediated neuroinflammation and axonal degeneration in dementia pathogenesis. And we also found significant associations between serum SCD-14 and worse cognitive outcomes. However, SCD-14 and also YKL-40, they are elevated in other inflammatory conditions as well. So some of these findings may reflect systemic inflammation-associated effects. And we found associations with a higher level of UCHLI and worse cognitive outcomes as well. And UCHLI is considered a marker of neuronal injury because of higher abundance in neurons, but it has been suggested that this also could mean compensatory expression of the enzyme in response to higher burden of misfolded tau and amyloid proteins. But its role in AD pathophysiology is still not clear, so future mechanistic studies are warranted. So we found similar associations in black and white participants. These participants were included in this study were mainly from North Carolina and Mississippi sites, so which fall in the stroke belt. So similar cardiovascular risk profile could explain this association in part. So overall, these blood-based biomarkers were not specific to Alzheimer's or dementia, but this may have, collectively, they may have potential to characterize individuals with multiple concurrent pathologies, and if further supported by future studies, they could be integrated with core AD biomarkers for more comprehensive staging and diagnosis of Alzheimer's disease as proposed in the recent revised research framework of Alzheimer's disease. So in terms of strengths, our study, the strengths include long-term follow-up. We did have data on both cognitive decline and incident dementia. We had comprehensive information on confounders. And in terms of limitations, we did not have information on blood biomarkers of amyloid and tau pathology for this subset of participants in the ERIC study. And also, we did not have information on the underlying etiology of the dementia. And finally, I would like to acknowledge my wonderful collaborators, without whom this study would not have been possible. Thank you.
Thank you,
Sure.
Thank you, Shrishti. Does anybody have any questions? Yeah, we have one.
I have 2 quick questions. First one goes to Hamilton. I saw like you have been trying to validate the biomarker of the ratio between the 2 proteomics and other conditions like FTD from GenFi. You might have already done this. Have you ever validated this like in Olink from UK Biobank, the two ratios?
Microphone.
Okay, yeah. So on Olink, actually, the YWHAG isn't measured. And they do have, I think, YWHAQ, but it's not as strongly associated. And so really the strongest signals we find are in SomaScan. And actually, we've also correlated with mass spec, and even there, SomaScan is slightly stronger, pretty consistently across cohorts. Yeah.
My second question is more, to the entire panel, and I think like from all those like univariate analysis by linking those proteomics proteins with other like biomarkers. What's your opinion about like using more advanced like multivariate like machine learning model to derive like some proteomic signatures that can potentially improve like prediction power, especially given the fact actually synaptic dysfunction has been super like implicated from both Hamilton's work and Kinnan's work. Thank you.
Yeah, I can go. So, it's definitely combining information from multiple proteins is going to be great for biomarker purposes. And I think the limitation is if we want to get at causality of which proteins cause Alzheimer's disease. I think that's where the single protein analyses from Keenan and many others are really useful, because there you're testing whether genetic variants change expression level of a particular gene and whether that's linked to a disease. And so then if you add multiple proteins, then you sort of lose the direct causal ability to test direct causal effects. But definitely for predicting outcomes in patients, multiple proteins is going to be the future.
Yeah, I echo that. I think for prediction, multivariate, for understanding disease biology, univariate has been the way we've approached it. I think in the multivariate models, relevant proteins can get kicked out of the model just because they correlate with something else that's a little more predictive, but may not be actually causal. So we always look at it both ways, depending mostly on our objective. I have a question for you, Srishti. So GFAP and YKL-40 were associated with cognitive decline and incident dementia, both astrocyte biomarkers. Were you able to put them both in a model, speaking of multivariate, and see if they contribute uniquely to dementia risk?
Yes. So they were not, I do have correlation coefficients actually, but they were not very much correlated when we put them all together. So the associations for GFAP and NFL as well as YKL-40, they hold. So even adjusting for each other. So
Okay.
yeah.
Good to know. Thank you. All right. Any additional questions? All right. Thanks, everyone, for coming. And I want to thank Carlos Cruchaga, Marilyn Albert, and Paul Worley for organizing this symposium. Thanks, everyone.
Thank you.